Okay, thank you for the introduction. Yeah, so I want to tell you today about this data set that we have been publishing just last month, and we call it Prenatal Human Connectome Patterns. So it is a multimodal MRI-based imaging data set of human fetal brain development at the mesoscopic scale. So if you were there yesterday and were lucky to attend Dr. Verma's great talk, you're already a bit familiar with this. So during the fetal life, and particularly the second half of gestation, which is outlined in red here, a lot of fundamental neurodevelopmental processes happen in the brain, such as neuronal proliferation, migration, axonal growth. Many transient structures appear and disappear, and this forms the basis for what the future brain will become. So during this time period, most of these processes have been classically studied with histology methods for a very long time. And these methods yield very detailed imaging in slices, but until very recently, they had quite sparse spatial coverage. On the other end of the spectrum, you have the recent developments of in utero MRI, and we are now able to image live fetuses in the uterus. And this is becoming more and more common, so that cohorts of dozens and soon hundreds of fetal brains are available. However, this remains limited to a macroscopic scale, and we have a large gap between those modalities. And this is where we want to work. We want to fill this gap with mesoscopic scale data of 100 to 200 micron resolution, and for that we chose to use ex vivo imaging at very high field. So using this kind of resolution and imaging methods, we hope we'll be able to tackle processes such as neuronal migration, development of the cortex, development of the fiber architecture, etc. Now, in relation to the existing literature on the subject, you can see here on the white background a review of the studies that use ultra-high field MRI to study the fetal brain. And you can see a trend where the higher the field strength, the younger the fetuses. And the reason for that is a simple technical limitation. Ultra-high field scanners that are commercially available have a very limited working space, and typically it is limited to a 5 centimeter useful diameter sample size. So with the method that we have developed in Neurospin, I'm going to present you just after that, we are able to overcome this limitation, and we want to tackle this period here, the late second and third trimester of gestation with ultra-high field imaging. On the bottom of the slide, you can see on the black background the histological studies, and until very recently, only paper-based book atlases were available with quite sparse coverage. And it's only with the recent papers by the Madras group that full brain, dense coverage histology has become available. Until now, however, they have been limited to the early second trimester, although I may soon need to update my slide. So, how did we overcome this limitation? Well, we are using a blockwise imaging technique. So, first, we image the whole brains or whole hemispheres, and we plan a cutting plane digitally. We then realize 3D printed cutting guides that we use to physically section the brain into separate blocks. We place these blocks in custom designed 3D printed containers because fetal tissue is very, very soft and difficult to work with. We absolutely need to minimize handling of that tissue. So for each brain, we design custom fitted containers, and the tissue blocks are sealed in those containers for the whole duration of imaging. After that, we can put the blocks into the scanner, but the blocks are generally too long to be imaged in one go, so we need to acquire multiple fields of view. Now to the actual scanning protocol. We are using an extensive scanning protocol with a standard T2-weighted anatomy at 100 micron isotropic resolution. We also acquire relaxometric data, so these are maps of the quantitative relaxation parameters for T1, T2, and T2 star. And we have an extensive diffusion imaging protocol with multiple B values and high angular resolution, which should give us access to the fiber architecture. Now that we have acquired the data, we have partial fields of view of the brain which we need to reconstruct. So I will not go into the details of that procedure, but we developed a full pipeline that is dedicated to this task. First, we have the intra-block registration between consecutive fields of view. This is a quite classical registration problem with overlap between the blocks. But the inter-block registration is a very tricky one because we have no overlap between the neighboring tissue blocks. So we developed a semi-automatic method with manually input landmarks that allow us to get a precise alignment of the sections. So at this stage of the project, we have done the first data release just last month. The data is available on the eBrains platform. So we have released 3 brains, which are typically developing brains without any known neurological lesion or malformation at 18, 27, and 31 weeks of post-conceptional age. And you can visit the eBrains website if you want to download the data. So these are the anatomical scans, but for each of these brains, we also have a wealth of different maps that we released. In total, we have 16 or 17 maps per brain, so I'm not going to detail all of them, but you can see here the relaxometry maps, and here are some maps based on diffusion imaging. All of these maps provide information on the tissue microstructure in terms of iron concentration, myelin, concentration of macromolecules, cell density, etc. And by analyzing them in combination, we hope to be able to characterize the microstructure of the tissue. So this is the 27 post-conceptional week brain, and you can see, let me play it again, you can see here that we have a junction line that is quite well handled by the registration, although it is not perfect in the immediate vicinity of the cutting plane. Now, using this data set, we can describe lots of features of the anatomy. Here, for example, is an excerpt of the cortical plate at 33 weeks of the gestational age. And you can see the complementary information that is brought by the different modalities. You can do the same thing on the hippocampus, and there the intrinsic 3D isotropic nature will really help to get a full 3D structural description of the hippocampus. And we also have preliminary results on diffusion imaging, where in this 18 post-conceptional week specimen, you can see the mostly radial organization in the cortical plate and the tangential organization of the superficial layers, and as well as the subplate. And we can use this kind of imaging to discriminate the transient compartment of neuronal migration from the ventricular zone to the cortical plate. Now, the work that remains to be done, well, we are acquiring additional specimens because we would like to get an even coverage of the developmental timeline during the second half of gestation. So more specimens are coming soon. We also have acquired histological data on the same tissue that undergoes ultra-high field MRI. At the moment with H&E staining, and we hope to be able to use it for doing correlations between the ground truth microstructure revealed by histology and the MRI contrast that we have. And yeah, so these are the perspectives for this work. We want to develop multi-parametric tissue signatures based on the multi-modality. We want to exploit the diffusion imaging to reconstruct tractograms because their MRI really has this unique power to delineate the fibers in full 3D. And we will do some proof of concept application to brains with agenesis of the corpus callosum to see if we can describe specific anomalies in these brains. I want to end by thanking the whole team that worked on this project, and in particular my student, Luca Acamon, who could not join today physically and will defend his thesis next month. And he was the one behind the development of the whole methods. And thank you for your attention.